Is my screen showing? Yeah.
Yes, it is.
Okay, great. Well, pleasure to be here. I'm Adam, and I wrote an essay about biomedical progress. A bit about my background, mostly in philosophy, but it became really interested in machine learning and molecular biology over the past couple years. And this is the product of that. So hopefully it will be useful for you all. Here's an outline of what I want to go over. So I'll do a brief tool review for an ML audience, explaining what's happened in the past couple decades of biology. Then I'll really briefly touch on current threads in the ML for bio space. Then I'll step back and we'll think about the biomedical problem from first principles. Then we're going to talk about some simple frameworks that might be useful in predicting and hopefully increasing biomedical progress. And then finally, it's going to get a bit more speculative. Yeah. So here's this useful taxonomy, I think, to understand biomedical research tools. So I call it the 3S taxonomy, and it says that all progress in biology, in the tooling we use to do biology, which is an experimental science, is in one of three categories: scopes, which like a microscope are used to read state from biological systems; scalpels, which are used to perturb biological systems; and simulacra, which are model systems that act as stand-ins that we can experiment and observe. So in case you've missed the past 20 years of biology research, which I mean, some aspects of this have been well publicized, like declines in sequencing costs. Other aspects have not. But long story short, a lot of these tools, particularly scopes and to a lesser extent scalpels, are on these exponential cost decline curves and increase in throughput curves. And so here I've given a few examples. So on the left, you have DNA sequencing costs declining from around $100 per gigabase in 2010 or so to around $1 in 2022. The figure's slightly incorrect. On the top right, you have single cell experimental throughput. So these are methods for prepping single cells to be assayed. And similarly, you see an exponential increase in throughput. And in the bottom right, you have the resolution of spatial transcriptomic methods. And a bit of background on that. So if you have a slice of tissue, say, not only do you want to know the bulk transcriptional state and lose all the spatial resolution, but you'd like to ideally know where certain cells are and what their contents are. And so we've seen huge increases in the resolution of methods that assay this, that can tell you not only the cell's contents at a very high feature resolution, but also where those cells are. And so these are all really exciting tool trends. And as a result, we're awash in data, and this is only going to become truer over time. On the left, you have the Human Cell Atlas, which is a consortium that puts together these large atlases of different human tissues and organs. And as you can see, there's not only an exponential increase in the size of these cell counts and data sets, but also the frequency with which they're added. So, to be clear, that this trend is super Moore's law. I mean, the pace is faster than Moore's law. In the top right, you have a single molecule fish. Oh, go ahead. Yeah. Oh, sorry, I thought I heard someone. In the top right, you have single molecule fish, which is showing how you can visualize the locations of RNA transcripts in tissues at extremely high resolution. And in the bottom right, you have a light sheet microscopy showing embryogenesis at very high resolution. to reiterate, there are these methods that are increasing in throughput, that they're decreasing in cost, that they're becoming more accessible, and as a result, there's just tons and tons of data to work with, which is super exciting. So while this was happening, or with a slight time lag, machine learning methods have come to biology. And so I just want to point out a few literatures, some large, some small, that might be of interest. So the first is single cell methods. I'd mark the start of this field, machine learning for single cell biology, with the 2014 pseudo-time paper from Trapnell et al. And so single cell analysis methods are concerned with things like fitting manifolds of lower dimensional representations of cell states, modeling trajectories, for instance, differentiation of cell types from one to another along these manifolds, predicting the fate of cells, including under perturbations, or genetic perturbations, things of this sort. And so this is a pretty well-established field. Single cell biology, as I said, single cell biology tooling has advanced tremendously. In a way, these single cell methods are on the forefront of machine learning for biology. So if you want to get into the field, I'd get up to date with that literature. Secondly, structural biology. Everyone probably knows of AlphaFold and open source variants of it. But very exciting work here done with geometric deep learning, equivariant neural networks, these sorts of things, which are quite promising. Probably less well known is spatial biology, but taking the spatial transcriptomic data I talked about before and actually modeling through space and through time the relationships of cells, which might reflect, for instance, cell-to-cell communication. Fourthly, representation learning on the genome and the epigenome. So I think you guys had a talk a week or two ago about predicting variant pathogenicity. So there's very exciting work on using transformers, for instance, to model sequence data. And finally, representation learning on images. So I'll note that UNet, an architecture that's the basis for a lot of the diffusion models we see nowadays, actually came out of biomedical image segmentation. Quite interesting. The point I want to make, though, is that biology tends to lag the state of the art in machine learning by roughly 3 to 5 years. That seems to be the case. So to give a couple examples, for instance, like you have, I'll give you some references to check out. So if you check out Fabian Thies's lab, he's doing great work on modeling single cell perturbational biology. Some really cool papers over the past year or two. But these are VAE based methods, which with some adversarial regularization thrown on to make your drug latent spaces a bit more interpretable. These are very cool, but they aren't revolutionary by any means. And if you trace them back, the work they're coming out of, it's like the VAE in 2014 by Kingman Welling, and some stuff about using adversarial regularization. It came around 2017. So roughly a 4 to 5 year time lag. In the middle, we have CLIP on the left, which Radford et al., which came out in 2021, and then you recently saw Hochreiter put out a very cool paper doing CLIP contrastive training with drug embeddings and image embeddings, which is cool. So it's roughly a 1 year time lag there. And then finally, a single cell trajectory modeling method from Krishnaswamy, Sumita Krishnaswamy's lab, which came out in 2021. And the basis of this was neural ODE, which came out in 2020, I think. And also it's based off continuous normalizing flows, which came out a year or two prior. Point of all this being, the machine learning for biology community tends to lag the state-of-the-art machine learning community by on the order of 4 to 5 years. And so it's worth keeping that in mind as we go forward. Though I do want to point out there are exceptions to this. On the left, you have UNet, which came out of, originally it was designed for biomedical image segmentation in 2015. To hear biomedicine is leading the state of the art. And on the right, you have a very cool receptor binding paper. And I add this because there's a lot of great research on geometric deep learning coming out of biomedicine. And in many ways, I mean, as we saw with AlphaFold, biomedical research is on the forefront of geometric deep learning. But I want to step back now. So we've just touched on a lot of different areas where machine learning's being applied to biomedical data, and I want to ask, what does this all amount to? What will this produce eventually? And you have all these different methods with the different inductive biases and different data sets, and it seems like it might be impossible to talk about all these in the same framework. But I believe that's incorrect. It's actually possible to talk about all these types of data and methods we use to model them with a pretty simple framework. So the dynamics framing of biomedicine is this. That there are only really two types of research. There's observational research, where you're trying to model the joint structure of biological data. And then there's experimental research, where you're trying to model what would happen if you do something, if you act on biology in some way. To just think of it as this observational and experimental. In my essay, I lay out a spectrum of approaches that can be taken to this dynamics modeling problem. And the two poles I call mechanistic mind and totalizing ML mind. So I'd say that the mechanistic mind is what biomedical researchers naturally bring to the domain. There are certain conceptual primitives they use. For instance, things like inhibition, upregulation, right? These are all terms describing biology at the mechanistic level. But I think these tools are indicative of a broader research ethos that they have, which I outline here. If you talk to a biologist, they aren't going to say, I want to control a biological system. They're going to say, I want to understand it. I want to model it in decomposable terms. Whereas the totalizing ML mind, as I'm calling it, is this. I don't think anyone yet truly thinks in these terms, but eventually we might, forego this sort of mechanistic decomposition and this legibility in favor of black boxes that simply predict and control biological systems without us understanding them. So one way to model this maybe is if on the left you have this extremely convoluted metabolic network modeling the inner workings of a cell, and even this is a simplification of what's actually going on. In the middle, you kind of have how the current machine learning for biology community thinks, generalizing here, but doing manifold fitting, modeling trajectories on these manifolds, and this is very reminiscent in some ways of how the machine learning community is thinking about modeling, and even as late as the early 2010s. And on the right, you have a black box. And perhaps biology will be going more in this black box direction in the future. So let's talk about what that might look like. So you might say, okay, we see where this is going. We're going to just have to stack more layers. Attention is all you need. That this will solve biology. Case closed. What more is there to talk about? Richard Sutton will be vindicated. Just throw more compute at it, throw more data at it, and eventually you'll have a perfect model of biology. But I don't think, and so you might call this view the scaling approach or the scaling hypothesis of of biomedicine, and I don't think this naive scaling approach will work for two primary reasons. One is what I'll call biocompute power, and the second is what I'll call active learning. That's the term that's used. So by biocompute, I mean what systems generate the data, the training data our models are fed. And so here you have a frontier of model systems where there's a trade-off between external validity onto humans, i.e. how well the model translates to humans, which is ultimately the system we're trying to target in biomedicine, versus the throughput cost or ethics or the regulatory hurdles associated with that model system. So in the top left, you have C. elegans, a very common model system. Could have put Drosophila there. Below that, you have some cells being grown in a dish, maybe induced pluripotent stem cells. To the right of that, you have a little kidney organoid, very nice looking kidney organoid, which is slightly more complex than the iPSCs, or cells just grown in a flat dish. To the right of that, you have a mouse. And finally, to the right of that, you have a human. And so the point here is that there's a trade-off that has to be made between the quality of the systems we can observe and experiment on, and how often we can do that or how expensive it is. And this poses a bit of a problem to just scaling up our data. So an analogy I make in the piece, well let me step back. So you've probably seen the graph on the left, I'm assuming, which shows this super exponential, supposedly decline in DNA sequencing costs, and compares it to Moore's law. In this naturally invites a comparison. You'd say, okay, maybe DNA sequencing is to the biomedical revolution as Moore's law was to the information technology or computing revolution. But I think that this is a mistaken comparison. So to return to our 3S framework, it'd be better to think of what I'm calling simulacra as the equivalent of compute power in Moore's law. So rather than sequencing, which is a type of scope in my framework, being the equivalent of microchips in Moore's law, originally memory, it's better to think of actually the direct analog of compute in biomedicine, which I argue are simulacra. And so you can model, I mean, this is a kind of a toy model, but you could say each model system we work with has a bioflop capacity, which is some product of its external validity onto humans, that is how well the findings from it port the humans times the throughput, right? And so therefore, if you really take this finding seriously, then you might say, okay, rather than further decreasing the cost of scopes and scalpels, so decreasing sequencing cost further, making CRISPR, for instance, cheaper and more accessible, actually the most important thing on the current margin to do to accelerate biomedical progress is actually to push this simulacra Pareto frontier, to return to the information technology revolution. In other words, you need to build more performant chips, make them cheaper, make them model your target system better. So that's the first objection to the naive scaling argument. Biocomputational constraints. As I just pointed out, this yields a pretty interesting, underrated area of research to look into. Secondly, why you can't simply add more layers or scale data and scale compute is because biology is an active science. We don't passively intake data from the universe as we might in, say, cosmology. Rather, we have to actively solicit data from biocomputational oracles, i.e. model systems in nature, right? And so there's a data selection problem here. And in the figure, this is from a recent paper on beyond neural scaling laws, where it shows that data pruning, which is kind of like active learning, can actually improve the information gain or the loss in or the decrease in loss in scaling per example, because certain examples are more useful than others at different points in the model's training. So in the piece, I outline what this might look like in the near term and the far term. In the near term, you know, AI agents probably aren't going to be selecting experiments. However, they could play an important role in aiding humans in doing experimental selection. In the longer term, we might see an interesting self-scaffolding behavior emerge, where AI agents are not only selecting experiments from their current repertoire that they have access to because they're given certain tools by humans, but they may actually start self-scaffolding their own tools in expanding their experimental repertoire and thinking over longer time horizons. So this is very speculative. So to return to the point of all this, there's a common narrative. It's pretty widely believed that the rate of biomedical progress has never been greater. In some respects, this is true. As I spoke about at the beginning of this talk, there has been unprecedented improvement in scopes and scalpels, and we're able to observe and perturb biology at higher resolution, throughput, lower cost than ever before. We must grant this. But this hasn't yet translated into similarly amazing progress in the downstream mortality indicators, for instance, that we'd like to improve. So it's worth pondering whether our current approach to biomedicine will ultimately deliver the sorts of biomedical improvements we have been promised. And so, I mean, the reason why I'm giving this talk to a machine learning audience is because I think machine learning will play an ever increasing role in this, and it's worth considering the incentives around the biomedical apparatus and how those may contribute to certain blind spots in answering this question. I urge you to think about this problem from first principles, look toward analogous domains in machine learning to possibly predict where things might go, and yeah, definitely contribute to the field because it needs much more machine learning talent. Yeah, that's about it. So happy to take questions, discuss.
I think there is already a question in chat from Cesar. I don't know whether he would like to ask it himself.
Yeah, absolutely. I actually added it in the middle of the presentation so as to not interrupt it. But basically, it's just to further the discussion on the point that biological sciences have a considerable lag after the state-of-the-art models. The question was, what would you attribute this lag to? Do you think framing the underlying biological problem as per state-of-the-art method standards ever a bottleneck in the process, or is it just straightforward regarding adoption in that regard, so to speak?
Yeah, that's a good question. If you were to compare, say, adoption of state-of-the-art methods in 2012 versus 2022, I think you're seeing faster adoption now. So ideas are diffusing faster. This is due in part to having more computational talent in the field, and also to Well, frankly, fashion's changing. It being acceptable to do these machine learning methods and biological. Yeah, I mean, this data, these large data sets didn't really exist even 8 years ago. So there wasn't really a possibility of doing or using machine learning methods to kind of naturally led to a lag. But I think this lag's being shortened over time as more machine learning talent moves into the computational biology field. Will they eventually reach parity? I mean, will the computational biology community start generating methods that are eventually adopted by the broader machine learning community? I don't think we'll see that, no.
I see, that's very insightful. The UNet example you provided particularly drives that point home, I think, and thank you for that.
Yep. Yeah, so I see there's a question about synthetic embryos. Yeah, so a bit of background. There are a couple labs. I mean, to this synthetic embryo news came out of the labs of Jacob Hanna and Magdalena Zernicka-Götz within the past month, I think. In the summary is they were able to use, well in one, there are multiple papers, but to simplify, they were able to grow mouse embryos to around day 8 of gestation, starting with only, in one case, only mouse embryonic stem cells, and in another case, embryonic stem cells and I think maybe trophoblast or some other sort of progenitor. Anyway, so this is relevant to the simulacra conversation, because the upshot is, hey, maybe instead of trying to tackle this extremely complicated developmental biology problem of trying to grow better tissues to model human biology, and trying to overexpress transcription factors and modulate other stimuli like mechanical cues and the nutritional composition of the media. Maybe we can just recapitulate development in toto using these synthetic embryos, right? And then use them to extract not only tissues we could do experiments on, but eventually tissues and organs that could be used therapeutically in humans. This is exciting. I think the Zernicka-Götz lab around a year ago achieved 7 days gestation, and now they're at 8 or so. So this is definitely an interesting direction. I would comment though that the yields of these methods were very low. We're talking sub 1%. The synthetic embryos were disorganized and dispatterned. They did form structures like the early heart, the neural tube. So the major developmental processes went on, but I'd express caution or skepticism that this approach will scale to generating useful experimental models at scale, and definitely not useful therapeutic tissues, at least over the short term. The interesting thing is these synthetic embryos will themselves act as useful model systems to better understand development, and then we can use this knowledge to develop more targeted, higher yielding methods for developing simulacra. Yeah, are there other questions?
I think there are a few more in the chat. Maybe William, would you like to go ahead?
Yes, thanks so much for the talk, Adam. Thank you, Nicolo, for organizing the talk. I find myself agreeing 100% with your intuition and what you described, Adam. Do you have any thoughts or ideas or guesses about what would the function be, or yeah, of those more black box models?
Ultimately, in the far future, we'd want these black box models to be able to take in any sort of biological data, different modalities, noisy data, incomplete data that describe the state of some system. Then we feed in some intervention that's performed, whether this is the encoding of a drug or a genetic alteration, and then predict the future states of the system across multiple scales. So at a high level, that's what we'd like. I mean, that would be obviously useful for therapeutics development. I think we're a ways away from that. And I'd also add that these models might not be black boxes eventually. There's some hope that they could be interpretable if we learn to probe them the right way. Yeah, so to one function of the black box would simply be as a dynamics model. Yeah. I see a question about, do you think physics/quantum chemistry simulations are going to play a relevant role as in silico simulacra? That's a good question. So some systems are more amenable to direct in silico modeling. Unlike even a single cell, which is incredibly complex, and we probably aren't going to model it bottom up at the molecular, submolecular level. It is possible that you could model something like, say, drug receptor binding directly in silico, modeling the quantum chemistry of this. I'm skeptical, though, that this will. I mean, suppose you had a perfect simulator for this sort of process, and you could work your way through drug encoding space to find the perfect drug to bind the receptor you want. Even if we had that capability, I still think that leaves most of the biomedical dynamics modeling problem unsolved. Knowing what to hit is and what that will do is an even larger problem ultimately than actually hitting the target. But I do think in silico simulation at this level will definitely play a role. I just don't know how much value it will add. Yeah, are there other questions?
I have one, if you don't mind. Could you go more in depth about what you see happening over the next few years? Because we've discussed in private about you not being particularly optimistic about the short and possibly medium term, which is something I agree about. I wonder whether you could, sketch out what do you see happening in the shorter term.
The one prediction I'm confident in is that the data flows will continue to have an exponentially increasing amount of sequencing data, of omics data, of spatial and imaging data. This is all grist for the machine learning mill. Now the question is, how valuable is this data? And that depends on the model system it comes from, if it's experimental versus observational. and to how useful is this data, and how high is the bar for actually making an impact on these downstream indicators of mortality, for instance. So if you buy the biocompute thesis and believe that small decreases in the external validity of these model systems require extremely large increases in the amount of data we get from these models to offset them, then I think you'd be pretty pessimistic. Even if we were to feed all the biological data we have access to into some large transformer-like model, it probably wouldn't be predictive in humans, one might argue. So conditional on not improving our model systems, I'm pretty pessimistic. That being said, over maybe a medium time horizon, as these data flows continue and the data accumulates, perhaps we'll simply have enough volume that certain that transfer just happens. So it could be that there's some transfer coefficient between a mouse process and a human process that depends on some kind of conservation metric that will simply work once you hit sufficient scale, right? I don't know what that is, but it's possible that if you just keep collecting experimental mouse data, eventually it will translate to humans. I think that the primary the primary metric that colors my pessimism is this biocompute question. Right? What is the conservation between mice and humans for these major disease processes that we care about, and how does that alter the scaling coefficients?
Thank you very much.
I have one more question, if I may. What would you advise aspiring machine learning researchers, as most people in the audience, I guess, to study or read more or practice more in order to contribute to getting to what you're describing faster?
Well, if you want to work in biology, then reading biology literature can only help to the extent that you don't let the field's ideas contaminate your own. I imagine as more machine learning people get into biology, you'll probably see the scaling crowd come with them, and resultingly this naive scaling approach tried in biological data, which will probably work to an extent. However, I think getting your hands dirty, understanding what biology actually is, will help give you better intuition for developing machine learning models and thinking of crafty ways to use data. Particularly to know where to look for data. Especially because the data formats vary widely. The data sets are not standardized usually. That they're all over the place. To the signal that's there, kind of tenuous. So having expertise in the domain and knowing how to think about the data and program in the right inductive biases might actually be really useful early on.
Thank you.
Maybe I can add some discussion as well. I think one very interesting thing happening recently is the improvement in our ability in terms of interpretability tools to handle even state-of-the-art models. I'm mainly thinking of the recent research happening at Anthropic, just a few days ago, Anthropic released a paper and blog post, which is absolutely stunning in terms of their ability to interpret features at the neuron level. I think in transformer models, although I think they're still relatively small scale, I think it might be possible that those models we train don't stay black boxes for long if interpretability tools continue improving the way they are currently doing.
Yeah, this does seem like a really promising line of research. Maybe the near-term future would look like feeding a bunch of data into these what we think of as black box models, doing this kind of interpretability work to spit them back out in human legible terms, and then humans reasoning over these representations, selecting the experiments, generating the data, feeding the data back in. So that's a potentially promising loop that could happen.
I posted the blog post I was referencing in the chat, by the way. All right. Any other questions for Adam? All right. I think then we can call it here. Thank you again, Adam, for joining us today. I will stop the recording now.